So today it's our an unusual day. It's a special day in the calendar for CVE, the Smith School at Cornell, for the Julian C. Smith Lectures. And this is our first time doing it online. Thank you for joining us. And it's a real pleasure and a privilege to have Dean Frank Doyle from Harvard joining us for this special event and a series of 2 lectures, one today and one tomorrow at the same time. So it's sad that this is our 5th year without Julian among us at his lectures. He passed away in September of 2015, and he's really sorely missed. He was always an extremely gracious and warm-spirited participant in these lectures, and his presence is again sorely missed. So Julian was a graduate of our school in a crisis period in the class of 1941. He got his bachelor's in 41. He got his master's of engineering in 42. He then worked for DuPont during the Second World War on teams related to the Manhattan Project. And in 46, he returned to Olin as a faculty member, and he held that position for 50 years. So Julian was admired at Cornell and globally for both scholarship and leadership. Most notably, he really put Olin Hall on the global map of chemical engineering with the authorship of an internationally recognized text, The Unit Operations of Chemical Engineering, with Warren McCabe and our own Pete Harriet. In 2008, Julian and Pete Harriet were recognized by the Warren L. Lewis Award of the AICHE for contributions to chemical engineering education. Julian also led our department through a critical transition during his time as director from 1975 to 1983. During this period, he navigated our school through a transformation from really a very much undergraduate focused department to one we recognize today with a pursuit of both excellence in education and cutting edge applied science and engineering. So it is this legacy that the J.C. Smith Lectures were established to celebrate. And it is in this context, or these contexts, that Julian would have been extremely pleased to welcome Dean Frank Doyle. So Frank Doyle comes to us from Harvard, where he is the John A. Paulson Dean and the John A. and Elizabeth F. Armstrong Professor. Like Julian, Frank is admired internationally for both his scholarship and his leadership. After a PhD at Caltech and a postdoctoral stay at DuPont, Frank rose through the academic ranks in chemical engineering at Purdue and Delaware. He then spent a long period in chemical engineering at UC Santa Barbara, where he served both as department chair and associate dean of research. In 2015, he became the inaugural John A. Paulson Dean of the School of Engineering and Applied Science at Harvard. So Frank, I dare to say, give him a title as a systems engineer. And he has made his name by taking on modeling and control of systems from many different areas: traditional chemical systems, electrical systems, biological systems, medical systems, and the list goes on. Not surprisingly, his honors come from a huge diversity of fields. He's a fellow of the International Societies in Control, Electrical Engineering, Medicine, and Science. And in 2016, he was inducted into the National Academy of Medicine. And I suspect other inductions will soon follow. So in his 2 Smith lectures, we will have a privilege of hearing about an applied endeavor related to the management of diabetes, that's today, and a more fundamental theme related to mechanisms of biological control, that's tomorrow. I hope you'll be able to join both, as I believe they will provide an impressive illustration of how he uses engineering approaches to probe basic science questions, and in turn, uses this basic understanding to drive engineering advances. So before passing the screen to Frank, I'll finish with a haiku. Frank, these haikus are a tradition of our department at seminars. And it's a haiku that's for both Julian and Frank. So here we go. Life's unit ops, biological clocks, controls makes good. So please join me in welcoming Frank Doyle and for the 2020 J.C. Smith Lectures.
Abe, thank you for that incredibly kind introduction, and certainly the first time in my career I've ever been introduced with a haiku. So let me see if this works. Are you guys able to see my screen?
Good, looks good.
You're good. Okay, terrific. Well, and some of you probably noticed the background I picked for my Zoom profile today, none other than your beautiful campus. I have to say, I was really looking forward to coming back to visit Cornell. It has been 36 years since I was on your campus. And I remember that because it was the Ivy League Sailing Championships in my junior year that we came up to Cayuga, and I had a chance to sail on what I recall were very cold waters. So I'm pleased to be able to represent Julian Smith through this lectureship. I read his bio, and many of the things that you mentioned, Abe, in that introduction were just amazing, truly inspirational. So I'm going to talk today about the work we do in diabetes, as Abe said in the introduction, and then tomorrow I'm going to lead you through more of a systems biology story. So today's story is really about biomedical control and the story of this artificial pancreas. So I'm going to start by kind of framing this in the context of understanding that I'm a control person by training, a systems engineer, as Abe noted. And for many of you, that may be nothing more than understanding how the thermostat in your house works. That's one way to approach engineering control. In my PhD and subsequent postdoc, I was working at DuPont. So the way I thought about control in those days was really more plant-wide control or refinery control, unit operations control, that perspective on control. But then a funny thing happened on the way to my academic career. I did a postdoc at DuPont, and I fell in with a bunch of biologists. And of all places, Wired magazine did this profile. I don't know if you can see my laser pointer here up on the top, neurobotics, biology meets computing. It was kind of a hokey name for the work we were doing. But the idea was that at DuPont, there was a group of us looking at how the rat's brain processed information and did homeostatic control. And those engineers at DuPont were trying to figure out if we could extract those principles and bring them over to the way that DuPont does process control. So reverse engineering, if you will. But of course, when you get written up in something closer to pop press than a technical journal, they tend to kind of get some of the details fuzzy. So for example, my discipline was labeled obscure, which was not very flattering. But my colleagues at DuPont, who were lifers in Wilmington there, they got an even less charitable characterization, portrayed as a lot of old guys who knew how to make Teflon without blowing up Wilmington. So nonetheless, this stuck, this sort of fascination with thinking about biological systems. And part of the reason it stuck can be seen in this slide here, where on the left, I'm showing you a circuit diagram of sorts. This is the gene regulatory map for a very simple organism, the sea urchin, and for a very focused part of the body, the endomesoderm, and for a very narrow slice in time during the development of this organism. So narrow in time, very simple organism, and a narrow slice of that organism's body. And yet you get this marvelously complex circuitry, over 40 genes involved in regulating that circuit. And this is, some of you may know, the beautiful work of the late Eric Davidson at Caltech, and his ability to map this out. And of course, on the right, I show you what an electrical engineer might think about as a circuit diagram, in this case, a shortwave radio circuit diagram. And what drew us to this field were the similarities in understanding how one achieves robustness out of circuits like this, out of systems that could do such marvelous control. And furthermore, how these circuits, both the biological one on the left, as well as the engineering one on the right, could accomplish that despite the fact that there was lots of noise in the system. Some of this was environmental noise or noise from neighboring organs or cells in the tissue. Some of it was intrinsic noise from things like stochastic gene regulation. But despite that noise, these circuits were able to achieve a level of robustness that by engineering standards would be enviable. And all of this was from a relatively simple biological circuit. So my lab has really focused on looking at how systems and control can play into what's often known as the iterative paradigm of systems biology. If we start on the top here, the idea of bringing computational approaches to understanding at a variety of scales, all the way from genes, proteins, networks, cells, all the way up to populations, using that to inform the design of experiments and the collection of high throughput data, interrogating those networks, using that data to then inform and update computational models, and going round and round this circuit with the idea that you converge when you are able to translate your priors, which you know from the literature, which you know from experimental databases, into novel biomarkers, targets, or therapies. That's sort of the paradigm in systems biology. And so our lab has worked at a couple of different scales in this. We've done work around Alzheimer's and understanding tau misfolding, and that's really been more at the network or protein scale. At the high end of the scale, we've actually looked at ecosystems. We've modeled coral systems and the marvelous synchrony of their spawning events. My talk, let's see, whoop, no, I jumped one ahead here. This is PTSD. This is an ongoing project we have right now with the military, in which we're trying to come up with novel biomarkers to classify individuals who've been exposed to traumatic stress. My talk tomorrow is going to focus on this scale, which is really looking at networks and cells in the circadian clock and the underlying gene regulatory control in those networks. But today I'm going to focus on diabetes, and in particular focus on the work that we've done on the artificial pancreas, which one could think of in this scale as being organs to organisms scale. And so to set the stage here, I'm going to just briefly introduce a paper that was published in the control community a couple years ago now, trying to identify some of the open grand challenges in the field. And this was a literal blue ribbon panel of who's who in the control field. And one of the big problems they identified was the problem of healthcare and how we move from our current approach, which is largely open loop. It's a manual intervention of the subject or the doctor without bringing feedback into the picture. If we could move that from open to closed loop, the argument they make is that we could really put a dent and really reduce the global costs of chronic disease, which in the forecast you see at the bottom of the slide there, over the 20-year period from 11 to 30, is forecast to be close to $50 trillion, so quite a sizable cost. And so for many of us who work in this field, there are a rich array of problems that are very exciting, whether you think about things on the left-hand side as replacing a natural organ with an engineered or a synthetic device, or you think about things on the right-hand side, which would be under a disease state, administering a drug or a therapeutic in some controlled manner. And of course, these are both very rich and vibrant fields, that of artificial organ design, as well as controlled drug delivery. So today I'm going to focus on diabetes, which many characterize as a global emergency, as well as a national emergency in the US. This is really a sobering slide in that it shows you the forecast over roughly the next 25 years till 2045, what the incidence of diabetes will be. And across the globe, it's going to grow by 50%. And in some of the really vulnerable parts of the globe, for example, in Africa or the Middle East, it's going to grow by more than 100%. So there really is a tremendous incidence here that's just going to grow with time and become a bigger problem. You can measure it in terms of the incidence. You can also measure it in terms of the healthcare expenditures. The first thing I would draw your attention to here is there's very disproportionate investment in healthcare, and that's really more in the first world parts of the globe that are investing much more in this. So in particular in North America, you've got roughly half of the investment, $377 billion were invested in the complications of diabetes in the year 2017. And that was about half if you sum up the rest of the globe here. So huge, huge investment. Well, let me dig into the problem a little bit. I'm going to start with due apologies to those of you who are on the phone who have formal medical background. I'm going to kind of take liberty with a figure out of a medical textbook here and show it in more of a conventional chemical engineering sort of process flow diagram. So this is the way that your body naturally does glucose regulation. The pancreas is functioning as a multivariable controller, if you have the perspective that I have. And there are two hormones which are managed, glucagon and insulin. So for example, after you eat food and your blood sugars rise, that increase in blood sugar will stimulate the pancreas to create insulin. When insulin then circulates, it does a couple of things, but primarily it allows the muscle and fat cells to take up that glucose, which brings the glucose level down. It reduces it. On the other hand, if your blood sugars are low because of exertion or stress or some other indicator, this will stimulate the pancreas to produce the counterregulatory hormone glucagon. And the primary role of glucagon is to break down glycogen reserves in the liver to produce glucose. And so this, of course, would lead to an elevation. So it's almost like your car having the brakes and the gas, where insulin is like the brakes, it slows things down. And the, glucagon is like your accelerator. It will speed things up in terms of the glucose concentration. And when everything is working fine, you get remarkably flat traces. So this is a blood sugar trace. And throughout the rest of my talk, I'm going to try to use this same scale, 0 to 400 milligrams per deciliter. And this happens to be one of my PhD students back in the day who wore a sensor for 24 hours just to show what an individual who doesn't have type 1 diabetes experiences in terms of blood sugar fluctuations. What you see is one of these rare kind of healthy flat lines in nature, right? Flat line we don't often associate with a healthy medical situation, but this is a remarkable tight control of the body's blood sugar. And I learned after we did this sort of protocol that this student had one of his typical 24-hour periods that included, in no particular order, pizza, beer, and bowling. So there were lots of challenges to his body, and yet his pancreas kept his blood sugars rock steady. So the version of the problem that we're interested in studying is type 1 diabetes. And in type 1 diabetes, the immune system attacks the beta cell in the pancreas, and so the production of insulin is impaired. Glucagon regulation is also affected, but not to the degree that completely suppression of the insulin production here. So now we've got a control loop which has been broken. And the solution, of course, as many of you know, if you have friends or family members who have diabetes, is this can lead to elevated blood sugars, so-called hyperglycemia. And the long-term consequences of hyperglycemia are numerous and include retinopathy, neuropathy, DKA, diabetic ketoacidosis. And so, of course, the solution is we put a human in the loop. That's the traditional way of treating type 1 diabetes. We let somebody self-administer their insulin. And of course, for the engineers out there, the chemical engineers who've had a process control course, we all know the problems with putting humans in the loop. They're inaccurate. Things don't often go according to plan. And so if we put a human in this loop, that can lead to challenges. The human may overdose the insulin, and that can give rise to immediate problems. Hypoglycemia, if we have a depressed level of the blood sugars, can in hours to minutes lead to complications that are incredibly dangerous. So it might start with headaches, lead to dizziness, coma, or even worse outcomes here. So that's the problem with putting in a human in the loop here. And here's an example of one of our subjects from back in Santa Barbara. This is right after I'd moved to Santa Barbara. I would call this a very typical subject, not one of our best, but by no means one of our worst. And this is a month-long trace showing you what their blood sugars look like over the course of that month. And what you can see here is there's a green zone. We're going to call this the euglycemic zone. Doctors know that if subjects can keep their blood sugars in that target zone, they minimize the complications of diabetes. But what you see with this subject is they spend all too much time elevated in a hyperglycemic state. And perhaps even more alarming, they spend a lot of time in a hypoglycemic state, including a lot of dangerous excursions below this literal red line where things are immediately consequential. So I'm going to show you just a quick tabulation of the data in that plot there. And this is a number I want to have you keep in your head for the rest of the talk. A typical subject self-administering can keep their blood sugars on track about 55% of the time. Slightly more than half the time, they're in the euglycemic zone. Just a little less than a quarter of the time, they're in a hypo state. And very close to a quarter of the time, they're in a hyper state. So that's a typical subject, self-administered, manual control, open loop control, however you want to think about that. And so the idea, if we go back to our cartoon, is get that human out of the loop and now put in automation. And that automation started for us with laptops in a clinic. It became tablets in a hip pack. These days, it's an embedded chip, which is either in your smartphone or actually in the devices themselves, that can close that feedback loop. And so if we think about the history of diabetes technology, it's actually a long and rich history. It goes back 100 years ago. We're coming up on the anniversary, the 100th anniversary of the first person who was administered with insulin. And over the course of time, particularly since the 70s, there have been remarkable advances in either mechanical equipment, computer algorithms, and concurrent with that have been remarkable landmark studies like the DCC trial, which was published in 1983. And then a number of agencies, including NIH and the JDRF, which got involved in supporting this in a very aggressive manner. The beginning of closed loop trials and a number of firsts, which happened through that period, including the very first fully automated clinical study in the US was done by our team in Santa Barbara back in the summer of 2007. If we flash forward to the present, or just a few months ago, we were published in the New England Journal with our longest study to date, a 6-month randomized trial. So things really have progressed remarkably over this period. And today, the diabetes technology is truly remarkable. It looks nothing like it did when those first devices came out in the 80s and 90s. These are slim, very tiny footprints on the body, remarkable choices for the subjects who are using diabetes technology. But still, for the most part, with a couple of exceptions, these are managed in an open loop manner. We're just beginning to see the first products, including this Medtronic 670G and a new tandem system that are coming onto the market in a so-called hybrid closed loop, meaning they control overnight, but around mealtime, you administer your own manual boluses. So I'm going to give you a very quick sort of snapshot of things we've been doing with clinical work, and then I want to spend the bulk of my talk talking about the algorithms that underlie our work. So we've been working in closed loop in a clinical or at-home setting for a little over a decade now. Back in 2007, I told you we closed our first feedback loop that summer in Santa Barbara, and we did this for only a few hours. And at that time, the blue graphs here denote the subjects that we've had enrolled. This is a cumulative plot. And the orange graph denotes the hours that we have closed a feedback loop on a human. And so in the beginning, it was on average 8 hours that first year. And you can see we've moved into periods now where these things are running literally weeks and months at a time. So let me just show you a peek into one of our trials and sort of what is involved. This is an ongoing trial that's happening in collaboration with Yale University. This is a pediatric cohort. So we've got very young children who are using this device. They're using it as a part of their phones. So these are not locked phones, but unlocked phones that can message and play video games and make phone calls. And our technology is an app, which appears on that phone. And you can see the kind of protocol that we follow here. This happened to be a 48-hour study. Over the course of time, the various interventions, whether it's activity or meals, that challenge the algorithm. What I'm showing you here are there's 3 different cohorts. There's adolescents, there's children, and then there's very young children. And if you look at the plot on the left, OL means open loop. This is what they do when they're not on the algorithm. CL is closed loop. And what you can see is the meal content went up. So actually during our closed loop trials, these kids were eating more and eating in a sort of, shall I say, uncontrolled manner compared to when they were in their normal open loop sort of therapy. So they really challenged the algorithm to try to keep their blood sugars under control. And this next slide has a couple different snapshots, including a video here, if I can get this to play. I think I have to take my laser off. Try this again. There we go. I don't know if you can hear that, but the kid's saying, "Mom, take a video of me." She obviously did. We had the benefit of getting it from her. But this is the kind of thing. These kids were doing rock walls. They were eating a remarkable amount of food, as you can see from some of these pictures, way more than they would normally. And yet, despite that, we were able to show, and this is data we just showed at a conference last month, we were able to show that we could lower their mean blood sugars, and we could increase the time they spent in that euglycemic range, even with these challenges. So that's just a peek into what a clinical trial looks like using this technology. But I want to now step back and kind of walk you through what the research look like as we evolved an algorithm that was safe enough to put in the human body with young children, for example, a pediatric cohort. So at the core of our algorithm is something called model predictive control. Model predictive control is a very ubiquitous algorithm in the control engineering field. It's used to control refineries, chemical plants, aircraft, robotics. Pretty much any engineering control application has been evaluated with model predictive control. And the roots of it go back to the chemical engineering industry, to petroleum refineries in the 70s, where it was first demonstrated. So without getting into all the algorithmic details, the main idea in model predictive control is that you have a model to predict. So we have a computational model that will forecast how blood sugars will change over time. That's going to drive a calculation that has to obey constraints. So for example, we can only put insulin into the body, we can't take it out. We'll feed that to an optimization code, which again, in our current use is a simple embedded piece of code that we put on the in an app on the phone. We have a function that we can use to evaluate how good the solution is. And altogether, then this algorithm will tell the pump what to deliver in response to knowing what the blood sugars are coming in. So we have our human here in this loop, that's our process, if you will, with the disturbances of things like meals, exercise, and stress. That'll lead to variations in blood sugar. The algorithm will work to calculate then the insulin dose that will try to keep this on a even keel, if you will. And so this is work that began roughly 25 years ago. I don't know if you can see on the screen, that's a 1996 paper that we published with Bob Parker, who was my student at the time. He's now a very successful faculty member at Pitt. And my dear colleague, Nick Pepis, who was with us at Purdue on the team. And of course, Nicholas is now at the University of Texas, and I've moved on to the next thing in my career. So early work. And a big part of what has characterized our latest work in this algorithm design has been customizing this, coming up with a tailored solution to the individual. So one way you can do that is take the attributes of the individual. So for example, what are their basal requirements, their requirements away from meals? What is their carbohydrate ratio, the amount of insulin to cancel a particular sized meal? Those become patient-specific attributes, and we've studied how to customize those. We've also studied how to use information to customize the algorithm itself, whether it's ranges that we put on the control bounds or adjustments of other elements of the algorithm that I'm going to walk you through in a few minutes here. Our latest work, though, combines them and says if we can get information from the subject, we should both customize their attributes and fine-tune the controller performance as well. So we just got FDA approval to begin a trial that I'm going to map out for you near the end of my talk here, but it combines the best of both to do this patient customization. And I would say the idea of customizing the algorithm was really early in our work at Santa Barbara. There were two doctors I had the great fortune to work with, the late Lois Jovanovich, who was a giant in the field, an expert in gestational diabetes, and Dr. Howard Zisser, who was also at Sansum in Santa Barbara at the time. Howard is now working with Verily on their diabetes technology. But we use the idea that in engineering, we could learn from patterns. So for example, some of our earliest work used something called iterative learning control, which is a very common technology in the chemical process industries. So the way that DuPont makes nylon, for example, in a batch is a very repetitive process. They make a batch, they formulate the nylon, they measure the properties, they go back and fine tune the batch parameters, and they run this all over again. A robot on an assembly line has the same repetitive pattern. Semiconductor processing has the same thing. So when you've got repetitive patterns, you can use something called iterative learning control to adapt or fine tune. Well, the observation for us was that 3 meals a day was a repetitive pattern. So if somebody with diabetes is eating breakfast, lunch, dinner in a very recurring manner, especially somebody who's disciplined with their management of diabetes. And so we could learn from that pattern, and just like one can do in batch chemical processing, we could come up with an algorithm that could improve over time. We could start in not so great control on day one, but after each batch, after each day, we could improve, and by about 10 batches, we've moved that subject into the euglycemic range. So that was our very first approach at doing this, and this didn't require continuous measurement, just required periodic or episodic data. Well, we moved into the realm of being able to measure blood sugar continuously. The technology advanced, and this was our first closed loop study with continuous measurements. It happened early around 07, 08, and at that time, the FDA had decided that they were going to regulate university research on closed loop algorithms. That set all of us back a little bit time-wise. So we went offshore, we went over to Israel and continued our trials in Israel, and ultimately married two cohorts, a Santa Barbara cohort and an Israeli cohort together. And if you remember the numbers I asked you to keep in the back of your head, you'll see here that during this closed loop, we were 70% of the time in range. 70% in that green band, the euglycemic range. We had virtually no hypos, less than 1%. And of course, the balance would have to be in hyperglycemia. So we were really excited because one might expect that even if we could replicate the average human and get 55% out of an automated algorithm, that would be a step forward. That would be automation. But in fact, automation let us improve that performance and raise it to an even higher level. The very next study that we published a few years later worked on further fine-tuning those attributes of the subject and got us into 75% time in range, about 1.25% or 1.5% hypo. This was an outpatient study. The previous study was in a clinic. So this one, subjects were out and wandering around. This is the streets of Santa Barbara. We had subjects in Virginia as well as at the Mayo Clinic. And what's really remarkable, if you look at this column, in the overnight window, we had 99% time in range. This is the critical window for parents with young children who worry about the levels of their blood sugar at night. And what you find here is that we're able to achieve remarkable control with virtually no hypos and no hypers whatsoever. We get very, very tight control. So let me dig into the algorithm a little bit and show you what it looks like. Here is the mathematics, if you will. Now, this is not your straight out of the box model predictive control that you would plug into a chemical plant or refinery. This has been carefully modified to be relevant for biomedical research. And so what we've done here is we have included things like the forecast of the glucose. We've put penalties on the insulin delivery. And we have an asymmetric cost function. And I think if I give you just the cartoon version of these things, it might be easier to follow. So for example, we tell the algorithm blood sugar should stay between these two bounds. That's actually one of our sort of unique elements of an algorithm compared to the rest of the community. We tell the algorithm that it's fine to live anywhere within these boundaries. And these boundaries can move overnight. For example, we could let overnight the trend drift up and then bring it back down during the day. We can do the same thing with the pump. We can tighten the pump control overnight and relax it during the day. We have asymmetric weights. That's reflecting the fact that hyperglycemia is dangerous long-term, but hypoglycemia is immediately dangerous. So hypoglycemia has to be avoided. So we can build asymmetric weights into the algorithm. And then this IOB refers to insulin on board. So insulin remains circulating in the body for hours. And because it's a drug that can lower your blood sugars, you don't want to stack the insulin, build up too much insulin circulating, so that one then gets a problem with too much insulin on board and potential hypoglycemia. So we build these as constraints into the algorithm as well. So with this combination of features, then we were ready to try this on a much longer trial. And this was a 12-week trial that we did, that we published just a few years ago now in 2017. In which we ran 24/7. The subjects were free living, open loop challenges, meaning whatever food, whatever exercise they wanted to have. They were a very tightly controlled cohort. So they actually started at hemoglobin A1C, which is the gold standard for the control of one's blood sugars. They started at 7.0, which is actually in a very healthy range. Despite that, we still brought them down to 6.7, which was a statistically significant improvement, all the while reducing the number of hypos. So SAP, I should have explained, that's a sensor augmented pump. That means you're wearing all the tech, but the loops aren't closed. So you can see your readout of your blood sugar. You've got a high-tech pump, but they're not talking in a closed loop manner. When you close the loop with the artificial pancreas, we really reduce those hypos. And again, to get to a 1% level on a 12-week study was really exciting for us to see that we could do that. But this was just the beginning of playing with the idea of looking at adaptation. And so now I want to describe the most recent trial that we published and then give you a preview into what the next trial looks like for our work. So we developed an algorithm we called a confidence index-based approach. And I have to say, I was inspired to do this by looking at the stock market of all places. So if one thinks about the stock market, and I think perhaps not the graduate students here, but the faculty online can relate to the idea of looking at your retirement portfolio and looking at the bands, the forecast on what the upside and downside of the market might be. So here's one of those typical kind of plots where if this is the present time, these bands depict high and low ranges on the forecast of the market. This is an optimistic forecast because it's still going up. It doesn't account for COVID-19. But what's important in terms of the work that we put together was looking at the volatility of the market. So this is a 5-year snapshot of something called the volatility index, which has made a lot of coverage in the press lately for obvious reasons. So for example, this was 2018, which surprisingly was actually a pretty quiet period. 2017, 16, 15. 2019 was sort of a bumpy period. Now, I gave a talk at a meeting in February, and I took this snapshot at that time. So remember this plot, and remember what 2019 looks like here, pretty noisy in this regard, because if I overlay today's data, I took a snapshot this weekend, what you see is those bumps here are virtually invisible because we're dominated by what's been happening in the last 3 weeks with COVID-19. So the volatility index is off the charts. Here's the insight. When the volatility index is low for my algorithm, I'm going to be aggressive with that algorithm and take very strong actions when I get signals from the measurement. When the volatility is very high, and volatility here might mean that my model's not predicting very well, or my sensor is faulting or having an issue, it's biasing. If I've got issues that reflect volatility or uncertainty, I'm going to back off. I'm going to be more cautious with my delivery of insulin. So that was the insight we were taking away from the market here, kind of a trust index, if you will. So here's the idea. We take that algorithm, and if I put up the chart here, suppose I've got a high trust index, meaning I'm confident, my model's accurate, my sensor's accurate, full steam ahead, the market's nice and calm. Well, if my subject goes hyperglycemia, then the algorithm's going to say, go ahead and crank on the insulin. I trust the calculation you're going to make. You're going to give me the right amount of insulin. If I go hypoglycemia, I will very dramatically sort of pull back and modulate the insulin, possibly letting some insulin in, but I'm going to be pretty aggressive with my design. On the other hand, if I have low trust, so my model is not accurate, my sensor's not accurate, something's off, I'm not making good predictions. Well, if I've got hyperglycemia, I may not respond quite as aggressively with the insulin. And if I have hypoglycemia, I'm going to be conservative and turn my pump off. I'm going to be very cautious. So you see the two extremes that might be induced by a measure that helps me understand what has changed with this subject. So we went and we implemented this in a 48-hour clinical trial. This was the paper that got published in 2018. You see the feedback loop here. We were running things on this tablet system with our app, the portable APS. We had a real-time sensor that could forecast every 5 minutes. And then we had the pump, in this case, that we were using through a Bluetooth relay to be able, the company is Insulet that produced the pump for this study. And what was really exciting about this trial is we got the time in range up to 88%. So 88% of the time we kept these subjects in range. That's to be compared to, for example, the time in range we had in the previous trials, which were in the 70s. We now got a remarkable boost in the time in range. And if we look at the data here, we can see they had very tight control most of the time spent in that euglycemic range. They started at about 75% when we began the trial. They finished at 88%. So this was huge. This really moved the needle quite a bit on what was feasible through an automated system. And I want to show you a chart that we have been collecting. We have an online database that we have made publicly available here that shows every single closed loop clinical trial over the last 6 years. I just picked that window because it already is a pretty busy plot. So what you see here is that every icon, a circle or a square, denotes a trial. The size of the circle or the square denotes how many subjects were in the trial. Big circle, big trial. If it's a square, that's a research group or collection of groups that use glucagon and insulin, so both hormones in their algorithm. Our work has only been single hormone, as have the vast majority of the studies, so these are insulin only in the circles. That trial I just showed you is this purple dot here, where we got up to 88%. What I'm plotting here is that time in range, 0 to 100%, or 55 to 100%, and then the time hypo, the time below 70 milligrams per deciliter. So of course the goal is to get up here, where you have virtually no hypos, and where you're maximizing your time in range. So you can see that a number of the recent trials, including our own here, are kind of nibbling around that periphery. But some of the larger trials, which often are pivotal trials to evaluate commercial technology, have larger, more complex populations and are less able to push the numbers in the direction that we're talking about here. So let me give you a preview of coming attractions. So this was an algorithm we published just at the end of last year. It was, in fact, a special issue of a journal to honor Nicholas Pepys, who was my original collaborator in this work, so highly fitting that we published this. And this was using a machine learning approach. So really one of the key underpinning technologies that underlie machine learning and AI approaches to try to learn from a long-term basis. And so if we go back to our feedback diagram here, inside the inner loop, we have the conventional artificial pancreas. We have a pump, a sensor, and our model predictive control algorithm that's going to compute, based on that information, what the optimal delivery to the subject should be. The innovation here is now this outer loop. And in that outer loop, we're trying to cope with long-term changes in that subject. So maybe it's the aging process. Maybe it's a disease. They get the flu, they get something else that changes their metabolic profile. And we want to be able to learn from the data to make modifications to our algorithm to improve the algorithm. So I mentioned earlier, the FDA, right before the COVID-19 business, back in late February, gave us a green light to advance with this trial. We're on pause with recruiting for obvious reasons, but here's the protocol. A subject would start week 0, and for the first 2 iterations, that's the first 2 weeks, there would be adaptation of their basal rates. This is the first parameter we'd adapt. If we can get our target achieved, the zone, then we move on. If we don't, then we have to try to adapt something else in that subject's characteristic, like their carbohydrate ratio. And that will do as many as 5 times. Once we clear this inner loop here, we can go to fine-tuning the components of the algorithm. So we can adapt components in the algorithm until we get a decent mean blood sugar. And so these loops will all be operating in the background until we get to week 12. So once a week, there'll be an adaptation. So for the folks who like the mathematics, Jeff Varner, a few others I know that are online there, here's the details. And so this is that machine learning estimation of what the response of the blood glucose levels will be. And the goal is that we want to try to get this into a nominal 120 to 150 zone right after the meal. So I'm showing you here one of the 3 components of the algorithm, the carb ratio. I'll spare you the other 2 late in the afternoon here, but just understand what's happening with the carb ratio. We try to optimize this, but then we put some limiters on the algorithm. So for example, we don't let that carb ratio change aggressively between updates. That would give us a non-physiologic kind of response. We also have bounds on what are tolerable average blood sugar levels. We have bounds on what we think are believable carbohydrate ratios. And then finally, we want to protect against hypoglycemia, so we have a safety constraint to ensure that we don't hit a hypoglycemic constraint. And so we intend to put this together. We've evaluated it in a simulator that the FDA has approved or accepted for evaluation of algorithms before they go into trials. And what you see here in the cartoon that's being animated, in blue are subjects that start out in a poor time in range, say 40 or 50%. And this adaptation algorithm moves them up to a very high, upper 80s, low 90% time in range. The error bars denote a population response. And then the red graph denotes the mean blood glucose, which starts at an elevated state, about 200. And over time, the algorithm does its adaptation and moves the subject down to a much more reasonable mean blood sugar level. So in that paper where we published these in silico analyses, We did a couple of different scenarios. In one scenario, we underestimated the patient's basals and overestimated their insulin to carb. This gave terrible time in range, about 40%, and we showed with significance that we could increase that to 88% time in range. We took another scenario where we overestimated basals and overestimated the insulin to carb ratio. This gave an interesting cancellation in that time in range looked okay, 75%. We still brought it up to 88, but that was because you were putting too much insulin in the body and you had a severe risk of hypoglycemia, 15%, which we rapidly reduced in a week or two down to near zero with statistical significance. So we've done our first step of evaluating the performance of the algorithm, and now we're about to recruit subjects to participate in the clinic. We're going to use that same app that I showed you with the wonderful children in the Yale Pediatric study. This is an unlocked cellular phone, so this is a big deal. It's the first time the FDA has let a phone, which is not locked down, totally controlled for clinical purposes, but just an everyday personal phone that merely has an app installed to do then the regulation, the communication with the pump and the sensor. And we've run a number of trials already where we've demonstrated that that app works very, very well. So as I wrap up here, I want to give you a preview not only of our next clinical trial, as I just did, but some of the other innovations that we think are going to be around the corner for the artificial pancreas. I mentioned a couple times that we're working on an app, which is a piece of code on your cell phone. We're moving this into the realm of an embedded control solution where we literally would have that algorithm embedded in a chip that might be resident, for example, on the patient's pump. So the microprocessor, all the processing, the RF, transmitting and receiving, all embedded in that system, communicating with the cloud, resident in the pump, which is then on the subject to give readouts and ultimately to administer their insulin delivery. This will let us get to kind of a goal we have of having an IoT, Internet of Things enabled, artificial pancreas, where now we can connect to calendars, activity trackers, the camera on your phone to be able to take a picture of a meal and have the food content estimated by an AI algorithm. We can build this into a cloud solution where we have the doctors able to access the information, the subjects access what they need, their self-care medical support team is able to access this as well. I think the big innovation for this is going to come in bringing new measurements into the scene. So we've begun to play with actigraphy measurements, so Fitbit and fitness devices. We're looking at meal bolusing using cameras and AI approaches to estimating food content, and then personalizing health records to try to get a better solution. But I would say one of the most interesting things, if I pause here for a minute and I ask the students out there who've had a process control course to think about this elaborate 25-year project that I've described to you is really a single input, single output control system. And when I first told my colleagues at DuPont about this, they said, really? Isn't that like freshman year, day one, feedback control 101, CISO control? And I said, yes, but you got to understand the human body is a little more complicated than a CSTR. Well, we're going to get to multivariable, and we've got this wonderful collaboration with Joseph Wang at University of California, San Diego, where he has some metabolic biomarkers that he's trying to prototype and build to actually do on-body measurements of additional hormones here. So we could add to our complement of having glucose, actual insulin measurements, which would be important, cortisol, lactate, other indicators that would allow us now to build an algorithm that could predict stresses and other components of the metabolic response. So that's the future. That's where things are moving, and we're very, very excited and energized by this. So let me wrap up by acknowledging the folks who did all the hard work here that I got the pleasure to talk about. The names in white here are graduates of my lab who've gone on to do great things in other places. The names in green are the members of my lab today who have been working in the lab. I mentioned a couple of my medical collaborators, including these consortia we participate in. The folks who pay the bills at NIH and these foundations, JDRF and the Helmsley Foundation, and then companies who give us medical devices we can work with. But the group that I'm really most grateful to are the subjects who've given us their time and their creativity and their energy. And so I want you to hear in their words what they think about this.
So the artificial pancreas is phenomenal because it will allow me, if I accidentally fall asleep early, to monitor my blood sugars throughout the night without me having to worry and get up. It'll mean when I'm working out and I'm dipping low, it'll be turning off my insulin. There's so many advantages with having a machine that's constantly monitoring you than you taking a break from your day and just keep testing and keep adjusting when the machine's helping you do it.
So that's Kirsten, who is one of our amazing subjects back in Santa Barbara. I'm truly grateful for all these subjects that we've worked with over the years. And finally, I want to thank you, the audience, for your patience listening to my talk this afternoon, and happy to pause here and I'll let Abe moderate some Q&A.
Thank you, Frank. So we can applaud, you can unmute and applaud for real, or you can use the clapping hands. So please put questions into chat, and I will help Frank navigate them. I'll give you a little moment. I'll start with a question of my own. So Frank, thank you. A beautiful illustration of a big, long effort. I'm interested to know, in your model within model predictive control, before and after you start to introduce the machine learning techniques, how mechanistic is the model and how much does it matter? And then as you look forward into a world of data-driven machine learning and so on, how does that answer change?
Yeah, so that's a great question, Abe. Thank you. So ours is not a mechanistic model. It turns out we have a time series model, which has some analogs to thinking about like a low compartment representation of the body, but it's a deceptively simple model. And what we have are universal dynamics, think plumbing time constants, but we have highly customized gains. So if you think about the sensitivity, how an insulin bolus introduced in the body would evoke a response out of subject A, B, C, and D, we can customize their gains, but their time constants are all roughly equivalent. And that has to do with the fact that the human adult body is going to have a very consistent volume turnover time constant that we don't need to customize. That part is quasi-universal. We fit the other components, and that's where the machine learning comes in to try to improve that regression. We might get one day to the point to be able to fit a PK/PD nonlinear mechanistic model. I haven't seen anyone show any convincing data because there's too many parameters. And again, we're measuring one thing. We're manipulating insulin, we're measuring glucose. And so it doesn't give us the fidelity to be able to fit a very complex model.
Very good, thank you. So we have some student questions. Tyler Dunbar asks, what is your perspective on open source DIY closed loop diabetic devices that some patients have recently been making and using without FDA approval?
Yeah, so this is the perennial question I get asked almost every technical meeting. I think there's some brilliant people out there, and over the years, I've worked with some brilliant subjects. I have a lot of PhD engineers who have type 1 diabetes that maintain spreadsheets and complex calculations to titrate their own insulin. They clearly have a personal insight that's hard to replicate by an engineering algorithm. And so I trust that they're doing good things. But Tyler, you put your finger on it. The FDA hasn't approved this, and it's risky. And there have been incidents in the DIY community with deleterious effects from these kind of algorithms. So it's a risk management question. And working where we are, we do need to get FDA review of our algorithms. We need to use those to bring them out. I think there's recognition in the community. There's something to be gained from the DIY approach, and how we harness that and build that into safer algorithms we can use with devices that get FDA review. That will be the challenge for our community over the course of the next couple of years, I think.
Good. Rachel Lacover follows up with, insulin can be expensive. How would you change your control loop to limit the amount of insulin needed?
Yeah, so that's a great question. We don't, we measure the insulin administered by the algorithm and track that and compare that to the total daily insulin a subject might use on their own. And in most cases, not every single case, but in most cases, we're able to reduce the insulin because there tends to be a underdose, overdose sort of thing. They overdose, they compensate with eating, and there's sort of this vicious cycle in a subject who doesn't have good control. When we can put our algorithm on, we really fine tune that and get that to just the right levels. So that very often is lower than the subject's own manual kind of intervention, but that's not the goal. The goal is to get the blood sugar stabilized, and we calculate then the insulin that can do that.
Good. So Abhishek Sharma asks, how does the location of insulin administration affect the ability to control blood sugar?
Well, that's a great question. Another one of my favorites. So I should have pointed out that right now in its present incarnation, we have a sensor which is subcutaneous, it's not intravenous, and we have a pump which administers subcutaneously. Of course, in the body, you've got in your portal cavity, you've got your pancreas squirting directly into the portal system, the insulin. So it's a very different transport problem. We have actually studied and played with devices which can either deliver directly into the portal cavity. We've done animal studies with an implantable version of the device. And the bottom line to your question, if I can get the insulin more quickly in, and likewise, I can measure that blood sugar more quickly, I take latency out of the feedback loop and I make the controller more effective. So the key would be to get these things in the body. The trouble is that requires an invasive surgery. Right now, these subcutaneous things are very simple to administer for the doctor to put on the subject. So, and the subjects then change their own sets. So it's very straightforward.
Okay, from Paula Eisner, can you say something about the relevance of this work to type 2 diabetes?
Yeah, so it turns out, type 2, by the way, is 90% of individuals with diabetes. It's the bigger problem for sure. And a relatively small fraction, the last time I was tracking this in the literature, I think it was estimated at 10 or 15% of individuals with type 2 do respond to insulin. They have some nascent kind of insulin sensitivity. The vast majority don't respond to insulin. That's the problem. You can't just modulate the insulin delivery. Their cells aren't listening to the insulin. That's the main problem with type 2. So there is a fraction of type 2 subjects that this algorithm would help. We have also begun to apply this algorithm to a very different application, which is post-bariatric surgery. So we can actually use our algorithms to administer glucagon in the case of somebody who's just had bariatric surgery and is recovering from that and now has dysregulation of their blood sugars. We can bring our algorithm to control the glucagon to normalize their blood sugar again. So there are a couple of different places where it might help, but I would say the vast majority of people with type 2 need a different solution than insulin. So these algorithms aren't going to help them.
Very good. Kui Wang asked, will food type and ethnicity affect the control? For example, vegans versus non-vegans, Western people versus Asian people who eat lots of carbohydrates, etc.
Yeah, so another great question. I glossed over the whole food content issue by talking about grams of carbohydrates. And we know that you've got fats, you've got proteins, you've got a mix of things that comprise that carbohydrate meal. And these different elements will be processed differently by the body. For example, fats very slowly. That's why something like a pizza is a biphasic response. The fats and the cheese are going to take longer to process, whereas the bread will be more quickly processed in the body. That gives rise to a very complex appearance in the bloodstream of the glucose, which requires a correspondingly complex cancellation by the insulin. We're just beginning to ease into the domain of looking at diet. We have we just published a study on pasta where we had a special glycemic indexed pasta that one of the companies had put out. We compared it to conventional pasta and white rice and showed where there were differences in the ability to regulate. But I'd say it's early days on beginning to touch diet. Most people are looking at very coarse approximation of food as a carbohydrate, when indeed, as your question connotes, it's much more sophisticated than that.
Good. Ziping Zhang asked, what's the size of your most advanced model? How many variables are taking care of personalizing that model?
Yeah, as I said, it would surprise a lot of people that it's a relatively simple, it's a third order transfer function is the way to think about this. It's a digitized version. So it's a Z transform for those of you who know what that is. And the dynamics, the pole locations, we fix across the subjects. We fit it lots and lots of our subjects and saw very consistent pole locations, i.e. dynamics. And I used that earlier, I think it was your question, Abe, when I talked about, you know, the dynamics of adults across the population are relatively consistent compared to the gains, the K value, if you will. Those become highly customized. So that's where we spend our time fine tuning. And of course, as we collect more data and have more subjects, we can also regress on the dynamic features. But for right now, we focused on that sensitivity parameter.
Good. Okay, I'm going to take two more, these last two questions. Chris Playson asked, this seems like a very good control structure for insulin for controlling diabetes. Do you see your control style, structure, and algorithm be useful for other researchers looking into other long-term managed diseases? Or due to specificity, should this algorithm only be used for this disease and treatment?
Chris, I really appreciate that question. Nobody's asked me that one before. So very much as we took an off-the-shelf model predictive control algorithm and customized it to the diabetes problem, I would say that a different application, for example, anesthesia control or cancer therapy, other controlled drug delivery problems can borrow some of the features of our algorithm, so a patient model, safety limits on the drug, but there will be others that will have to be highly customized. The regimen, the time scales, the sampling rates. So some of it can be inherited and built into an algorithm for a different disease, but much of it will have to be customized to that particular problem.
Yifan asks, what approaches would you take to further improve the responsiveness of the algorithm so that one can further reduce the time needed to correct the initial over or under prediction of baseline?
Yeah, another great question. By the way, Abe, on behalf of your audience here, I have to say I always judge these talks by the quality of the questions, and these questions have been excellent. I really, really have enjoyed this. So I'll give you two answers, Yifan. So one would be going inside the body. If we can remove those latencies of getting from the subcutaneous depot into the venous circulation and take that time lag out, we will do much better at blunting the postprandial, the post-meal excursions. So that's one step that we absolutely have to do. The other thing, I didn't go into the details when I showed you my charts, but the hardest meal of the day for us is breakfast. It's not because it's the biggest, it typically isn't, but because the body's circadian response leads to fluctuations in the insulin sensitivity that make it more resistant. You got to pump more insulin in to cancel that breakfast meal. So what I want to do, and this is future work that we're just beginning to kind of map out, is build part of what I'll talk about tomorrow, the circadian clock, into this algorithm so we can customize time of day and now have an even more tailored metabolic response. We can account for things like dawn phenomena.
Very good. Well, please join me in thanking Dean Doyle for an excellent first J.C. Smith lecture, and please join us again tomorrow. Thank you.